All right. guys. Classic. I always let the intro run way longer than I want to. Just bear with me for 1 sec while we futz around with some settings, get some things working here. All right, Bob's your uncle. We got a webcam. So, what are we going to talk about tonight? Last week we were discussing some of the science around how vaccines work and proteins. So I thought I'd just start with a quick little recap of some of the subject matter we covered, and perhaps actually, just guys, give me 1 second here. Just got to check something. Sorry, my cat just lost his mind there. Had to make sure everything was okay. Oh, right. Anyway, so quick little recap. Great start to the stream today. I know, classic. What are proteins? How are they made? What is DNA/RNA's role in all of that? And then we're going to talk about a little bit about the protein folding problem, AlphaFold 2, and hopefully segue into some of the mRNA vaccine science, how that stuff works, the lipid delivery mechanism, the actual mRNA mechanism itself, talking about some traditional vaccines and how those work. So yeah, please participate. Let me know what you guys think as we go deeper. To get started, there's a really awesome video that I enjoy particularly, called Why Do Acids Burn? And it covers a variety of topics that we need to get to. One of them is the way sort of molecules work, which is atomic bonds and some of the properties of those, and why acids are particularly good at dissolving or causing those bonds to break. Talking a little bit about the science behind proteins, giving us some good visual explanations of those things, and that's going to help us kind of segue into how proteins are synthesized inside of cellular biology. So hopefully we'll look at some cells, we'll look at how those work, how those synthesize proteins from DNA and mRNA, go through the whole process. So I'm going to get this video started, and then we're just going to dive right in. Here we go, guys.
I had this mild fear that one day in a sinister evil laboratory, a mad scientist who concocted a super acid would one day accidentally spill it on the floor, and it would begin its long journey of eating through the earth. This is of course not possible. The chemical world may have different rules than our world, but it still yearns for the same thing: stability. The reason it's easier to sit rather than stand is the same reason wood burns and metal corrodes. The end result is more stable and takes less energy than the initial state. Basically, just like us, if chemicals have the opportunity to make their lives more easy or stable, they'll take it. But what dictates the stability of the chemical world? The stability of a molecule relies on the strength or energy within the bonds holding it together. These bonds are formed from electrostatic attraction between protons and electrons, and just like with magnets, this attraction gets stronger the closer they are together. Molecules are most stable when all the electrons are as close to as many protons as they can be. However, there are a ton of factors or rules that dictate how close electrons can be to specific nuclei of atoms. The most important of these factors is called electronegativity. As an atom increases in size, it also increases the number of electrons orbiting around it. Because electrons have like charges, they don't like to be near each other.
So if you ever played with magnets and you've ever tried to stick two negative ends of a magnet together, what are they going to do? They repulse. Or two positive ends are going to repulse from each other. That is the magnetic force in effect. We feel that. It's not necessarily the magnet. That's that's our sensation, our perception of the effect of the magnetic force, and that exact same thing is happening on like the atomic level as electrons float around protons and neutrons.
If enough electrons begin orbiting our nucleus, eventually the attraction of the nucleus isn't worth the repulsion of all these electrons. at which point new electrons start accumulating in another outer shell. This is important because now every new electron that is added not only feels the attraction of the protons in the nucleus, but also the repulsion of the electrons in between them. Electronegativity can be thought as the ratio between protons in the nucleus and the electron shells underneath the valence shell. It's not exact, but it fits pretty well. This means that these elements have higher proton to electron shell ratios, meaning electrons, if given a choice, would prefer to spend their time here. So then, what are acids? For this, I'm going to stick to the most common and most prevalent description of acids and their behavior. The Bronsted-Lowry description states that an acid is a molecule that can donate a proton or hydrogen atom. What does that mean? An acid is a naturally unstable molecule. The more unstable it is...
So, if I'm not mistaken, I believe this is hydrofluoric acid, hydrochloric acid, hydrobromic acid, and I'm not sure which one that is. This is iodine? I wouldn't even know how to pronounce that one.
The more acidic or corrosive it is. Just like you and me, acids don't want to be unstable or unhappy. So, if given the opportunity, they'll react with something to change that.
That's the nature of an acid, is that it has there's a property in which it can donate a proton or a hydrogen to something and cause those bonds to break. And he alludes a little bit in what like bonds are and how important they are in chemistry, obviously.
But why are acids unstable? If we look at the most common or abundant acids, we see a trend. A hydrogen atom bonded to an electronegative atom. Now remember, electrons want to be as close to as many protons as they can be. That's how we form the most stable molecule. So if we imagine the point of view of these electrons bound to the hydrogen, they may be thinking something along the lines of, why am I hanging out with this loser? Just give me any excuse to ditch this guy so I can go hang out with all those Chad protons over there. But these electrons have a responsibility to hang out with Harold Hydrogen, and they can't ditch him for no reason. So this bond isn't very strong. However, if another molecule shows up with another pair of electrons to hang out with Harold, then these electrons will go, oh hey, have you met Harold? He's really good. Well, he's your problem now. Bye! This is essentially what acids do. They exist in a constant state of instability, always looking for a lone electron pair to pawn off their extra hydrogen to stabilize themselves. If the person they can pawn off their hydrogen to is more stable with it than the acid, well then they do it.
There you
Hence, strong acids are very unstable and can force almost anyone to take their hydrogens. Additionally, a single molecule of acid can only burn another single molecule. So that's why acids don't just keep burning through the earth. Eventually, it runs out of protons to give. But why do acids burn us? A chemical and thermal burn produce the same result. They destroy proteins. Proteins are the most important biological molecules in our bodies. They do essentially everything that keeps you alive. The reason they can do these things is because they are perfectly and exactly folded in a very precise shape. This shape is maintained by intermolecular forces and interactions, the strongest of which are salt bridges and hydrogen bonds. Now, I won't go super chemistry on you, but every amino acid, the things that make proteins, has a carboxyl group, which is...
So we're going to be coming back to this in a little while, not specifically to look at every single amino acid, but kind of to understand why amino acids are important just in the process of cellular biology.
The acid part of amino acid. But in proteins, the carboxyl group is often missing its acidic hydrogen and thus has an abundance of electrons and a negative charge. This negative charge can then bond ionically to an amine ion of another amino acid, or hydrogen bond with an alcohol of some other amino acid. These interactions play an important role in keeping a protein folded together. However, if we introduce an acid, this abundance of electrons on the carboxylate ion is a prime location to unload the acidic proton. So this carboxylate ion receives the acidic proton, becomes a simple carboxyl group, and loses its negative charge. If enough acid reacts with enough ions, the forces holding the protein together will fail, and it will unfold into a new shape. Without its original shape, it can no longer do its job, and if enough proteins become corrupted, then the cell becomes a useless mass of organic tissue, a burn. Bases do the same thing, but in a different manner. Instead of donating a proton to the carboxylate ion, they rip a proton from the amine ion. However, unlike acids, most bases have a hydroxyl group that will turn into water after the reaction. This water can then work with the base to start hydrolyzing or splitting other organic bonds via alkaline hydrolysis. So theoretically, if you needed to dispose of a body, a strong base is the better option, as it will be a little faster for dissolving some components. Lastly, how can we store these caustic substances in simple plastic containers? Well, remember, it's all about the electron's happiness. The simplest plastic, polyethylene, just looks like this. Now, if you were an acid, there's no free electrons to take Harold away from you. You'd have to rip apart a bond to get rid of Harold, which is possible, but only by the absolute strongest of acids, and if given some heat energy to help loosen up those bonds.
So that's another point too, is that like if you get an acid spilled on your hand and you experience like the sensation, the pain of burning, you also experience like thermal heat, because there is a chemical thermal reaction occurring on your hand. But again, because acids are highly reactive, they react with other molecules to break down their structures.
The same thing happens with bases. None of these electrons would be happy giving up their proton to the base, so there's no reaction. Thus, almost all acids and bases don't eat through plastic, but have no issue burning us.
Got my water, one sec. Really highly recommend checking out this guy's channel. It's great content that I particularly enjoy watching and talking about on this channel with you guys. Really quickly, since we talked about, or we just watched a little bit on how proteins are destroyed, let's try and find how does a cell create proteins. Let's see if we can find something cool here. Let's take a look here. Okay, let's try Mr. Biology 360. Sounds great. Oh, thank God I have a limiter. Jesus Christ, that was loud.
Welcome back, guys. Okay, today what we're going to talk about is a simple review that will help.
This is the video I want.
Hey, it's Professor Dave. Let's talk about DNA transcription and translation.
He knows a lot about the science stuff. Professor Dave explains.
Great, but before we get into the DNA transcription, we need to understand what the structure of a cell is very quickly. So, structure of a cell. Let's look for something fairly. Yeah, Khan Academy is great. Let's do a Khan Academy.
Let's talk a little bit about the structure of the cell. I've done a lot of videos where we deal with things that go on inside of them, but I haven't done one where we just talk about the entire structure of them. So a good place to start is, let me just draw the membrane, and the cellular membrane is a good place to start because this is what separates the cell from the outside.
So we're going to try and skip through some of this because I hope that you guys have had like a basic biology class in your time. If you haven't, feel free to let me know in chat. If you do want to watch all the details here, go through all the processes in cellular biology. But we're going to try and skip ahead a little bit. Some of this stuff, I am not going to lie, like I'm not super up on all of the differences and the finer details here. But the major point, or the major pieces that we want to talk about here are, Actually, you know what? Let's just go through the whole thing, guys. Why not? It's only 20 minutes. It won't take that long. And we can skip through the boring parts, okay?
Talk about the entire of DNA inside of it, and I won't go into the details of how DNA defines what an organism is. I've done that in
We'll
some
get
detail on the
into
DNA
DNA
videos,
later,
but
don't
all
worry.
cells have DNA here. This is more of a kind of an anatomy of a cell video than necessarily the function, but we'll go into the function because we need to know what these different parts do. So this is right here. This is the DNA. This is the DNA, and it's in its chromatin form here. There's also little proteins here. There's not in all organisms, but we're going to stick to eukaryotes, and I'll talk a little bit about the difference between eukaryotes and prokaryotes in a second. But you have DNA. As I've drawn this cell right now, this is pretty much any cell. Any cell in any animal or plant or whatever kingdom could look like this. I haven't drawn a lot of the details. I've just drawn the DNA and the cellular membrane. Now, here's kind of the first major division in the living world, or at least from our point of view, or that, you know, it seemed obvious, is that some cells have a membrane around the DNA. Around the DNA. So they'll have a membrane around the DNA that separates the DNA and the chromatin and everything that makes up the stuff within the DNA, separates that from the rest of the cell, and this is called a nucleus. This is called a...
The nucleus is the powerhouse. It's not the powerhouse of the cell. It's actually the mitochondria, technically. But the nucleus is like this, where all of the information on how the cell is supposed to reproduce is stored.
Nucleus. And I said that's a major division because when some people looked at some cells and they saw nucleus, and other cells and they didn't see nucleus, they say, hey, this is a good way to classify organisms. So they called things that had nucleuses eukaryotes. Eukaryotes. These have a nucleus. So as I've drawn this cell right here, it is a eukaryote. Now, if you do not have a nucleus, you are dealing with a prokaryote. Prokaryote. No nucleus. And examples of prokaryotes, the two big groups of them are bacteria.
Interesting.
And archaea. And archaea are really interesting. We know very, very little about them. They were originally thought to be types of bacteria, but now people are realizing that they're this whole completely other group, and we've actually observed a very small subset of them. So it's a very fascinating group. And it actually turns out that evolutionarily speaking, you shouldn't make this division first, that it actually makes more sense to divide things into eukaryotes, I'll just write that, euc, bacteria, and archaea. That you don't want to do this division first, that there are actually three separate groups that you want to start off with. Archaea. But we'll do that. We'll talk more about this in future videos. But if you want to say who has a nucleus, well, eukaryotes have a nucleus by definition.
Nice.
Who does not have a nucleus? Well, the bacteria and the archaea do not have nuclei. It leaves the nucleus, and it gets translated into proteins at the ribosomes.
So this part's really important. So we'll talk about the mRNA vaccine in a little bit. This is the process that the vaccine is using. It's using ribosomes to have mRNA transcript into a protein. So we'll get into the details a little bit deeper here, but...
So the ribosomes are these little complexes that could be floating all over the cell, and we'll see in a second that they can also be attached to these other membrane structures. So this is a ribosome. Ribosome. And if all this talk of DNA transcription into mRNA and mRNA leaving the nucleus and traveling to the ribosome to be translated into proteins makes no sense to you, there are several videos where I go into that in detail. But what I want to do is just focus on all of the different parts to kind of give a big picture of things. So ribosomes are where mRNA that gets transcribed inside the nucleus from DNA, where it gets translated into proteins. So you can kind of view them as the place where information actually turns into proteins, which then can be used anywhere else in the cell. Now, one interesting, and these ribosomes, they're made up of proteins, and actually they're made up of RNA. And so one question is, well, where are the pieces of the ribosomes made? Well, some are made by proteins, which might be made in other ribosomes. But some of it, the mRNA ribosomes, you can kind of view them as this big, you know, this mess if you were to look at detailed. There's some protein there, and I'm not drawing it that realistically, but then you have some mRNA tied in with the protein, and the mRNA isn't used for actual information purposes like it normally is when it goes from DNA to the ribosome. Within the ribosome itself, ribosomal RNA is actually used as part of the structure. It actually helps the ribosome function as a ribosome.
Interesting.
So it's part of the ribosome.
So the structure of that protein contributes to the overall like structural integrity of the ribosome. It's fascinating.
All of that gets built in a part of the nucleus called the nucleolus or the nucleole. Let me write that down. So
I think this is actually as far as we really need to go, at least in this part here. So the most important thing that I wanted to touch on was the process in which proteins are created. So we're gonna talk about transcription. That's going to introduce the mRNA aspect of things for us.
Now that we understand the structure of DNA, it's time to understand exactly how this molecule codes for a particular organism. How is it that a single cell containing a specific set of genetic material will result in the development of a fish, or a cat, or a human? To understand this phenomenon, we have to learn about transcription and translation. This is the collective process by which the genetic code is read by enzymes in order to produce all of the proteins in an organism. A chromosome is a very long molecule consisting of many millions of base pairs. Most of these bases don't do too much, but certain portions of the chromosome are special. They are called genes. These are the parts that code for different things. In a human, a gene will be on average around 10 to 50,000 base pairs long, though the longest is 2.5 million base pairs. And when a gene is expressed, a specific protein is produced. So how does this work? The first step is called transcription. This is the process by which enzymes use one of the strands of DNA within a gene as a template to produce a messenger RNA, or mRNA. To do this, RNA polymerase, with the help of proteins called transcription factors, binds to a specific sequence within the gene, which is called the promoter. And
which is just a protein. That's all you guys need to keep in mind. Don't get hung up on the terminology. This is just all of these are different types of proteins that your body naturally produces from your DNA, right? Your DNA has the instruction sets for these things, okay?
Prise the two strands apart. One of the strands will serve as the template strand, or antisense strand, meaning it will be used to generate the mRNA. And the other is the non-template strand, or the sense strand. RNA polymerase doesn't need a primer. It simply initiates mRNA synthesis at the start point and then moves downstream along the gene in a process called elongation, synthesizing the mRNA as it goes, reading the antisense strand from 3' to 5' and generating the mRNA from the 5' end, attaching RNA nucleotides to the 3' end as it goes. This is very similar to the way DNA polymerase synthesizes DNA as it moves along the template strand. The main difference here is that RNA is being synthesized, which, as we recall, will be ribose rather than deoxyribose, and it will have uracil instead of thymine. Unlike replication, RNA polymerase zips DNA back up as it goes, keeping only 10 to 20 bases exposed at a time. Once RNA polymerase reaches the end of the gene, termination occurs.
So the best way to think about this is like a zipper, okay? So your DNA is when is zipped up, and what happens is when a particular gene needs to be expressed, some chemicals find that spot on the zipper with the instructions, and it causes the zipper to unzip. And then what happens is it sort of codes to base off of this, almost like a key, right? Like a key is set off by the shape and the size of the teeth. And so it codes off of that key and then rips off. And then that RNA goes and gets used to actually create the protein. And then the DNA folds back together. So it doesn't destroy the DNA. It just opens. It's almost like opening a letter, reading that tiny part of the instruction that you need, and writing it on another piece of paper so that you can go and do something with it.
Enzyme detaches from the gene and the DNA is returned to its original state. But we have produced an mRNA. This carries with it the information encoded in the gene, and after a few quick modifications during RNA processing, it will leave the nucleus, where all the genetic material, or chromatin, is, and move into the cytoplasm, where it will find a ribosome. This is where translation occurs. During translation, the mRNA acts as a code for a specific protein. This happens because each set of 3 bases on the mRNA, which we call codons, will code for a specific anticodon, which will be carried by a specific transfer RNA, or tRNA. And each different tRNA is covalently linked to a particular amino acid.
So, I know this might be getting a little, like, for those of us who aren't biology experts or didn't do well in biology, these are essentially just chemicals. A, U, G, U, G, C, A, G, U, C, C. And kind of like magnets, like they have an attraction to each other. They bond to each other, and the opposites attract, right? And this acts as almost a coding layer in order to sort of say, okay, well, if there's an A here, then I want a U here, and UA bends to MET. This is how it built, or UAC bends to MET. So this is sort of the creating the instruction set almost by writing it backwards in a weird way. Now, I wanted to go back to... We're looking... Where's the acid video that we were just watching?
When I was a kid, I had this
So, if we go back to...
fear
I think it was...
they'll react.
Stop talking, please. Here we go. These are the amino acid groups, right? So when they're talking about the different groups, these are the chemicals, the different amino acids that synthesize proteins. So these little chemicals are given instructions on how to not only click together, but how to fold together. And we're going to get into folding and the whole protein folding problem shortly.
The nucleotides into these codons is called the reading frame. Since there are 4 bases and each codon has 3 letters, 4 cubed gives us 64 different possible codons, which is more than enough to code for all the amino acids we need. Here is a table of all the mRNA codons and the amino acids they code for. Notice that there is some redundancy, with multiple codons resulting in the same amino acid.
Interesting.
But there is no ambiguity. Each codon corresponds to a particular amino acid. Notice also that some of these codons are special. AUG is the start codon, which initiates translation by coding for methionine. And these 3 are stop codons. These are the ones that terminate translation. Translation will occur inside a ribosome. The small ribosomal subunit binds...
This part's important when we start talking about how the vaccine actually manufactures the proteins.
...to an mRNA and an initiator tRNA, which adheres to the start codon. Then the large ribosomal subunit joins to complete the translation initiation complex. Then the tRNA that corresponds to the next codon after the start codon will enter the ribosome. This will carry with it an amino acid, which becomes covalently bound to the methionine from the initiator tRNA. The first tRNA detaches and leaves the ribosome, which has shifted over, making room for the next tRNA. The new amino acid links to the first 2, and this process continues all the way down the mRNA. As tRNAs enter and exit the ribosome in a sequence that is dictated by the codons on the mRNA, a polypeptide chain will grow. This continues until a stop codon is reached, at which point the completed polypeptide will swim away, most likely entering one of the cell organelles for folding and further modification. So in this 2-step process, DNA is transcribed into an mRNA, and then this mRNA is translated into a protein, all simply by obeying the base pairing that occurs in nucleic acids. And since every gene codes for a specific protein, and proteins make up most of what you are, from your muscle tissue and organ tissue to all of your receptors and enzymes, this is how DNA carries the code for a living organism.
Pretty cool stuff, guys.
Thanks for watching, guys. Subscribe to my channel for more tutorials, and as always, feel free to email me, ProfessorDaveExplains@gmail.com.
All right, so now we know a little bit more about messenger RNA and proteins. I want to quickly introduce you guys, though, to the protein folding problem, which I think is an interesting. We're going to watch Lex Friedman video on AlphaFold 2. I think it was this one here.
I think it's fair to say that this year, 2020, has thrown quite a few challenges at humans. So this is definitely a big leap, but it's also fair to say that the internet is full of hype about this breakthrough. So let me indulge in the fun a bit. Some of it is definitely a little bit subjective, but I think the case could be made on the life science side that this is the biggest advancements in structural biology of the past 1 or 2 decades. And in my field of artificial intelligence, I think a strong case could be made that this is one of the biggest advancements in recent history of the field. So of course the competition is pretty steep, and I talk with excitement about each of these entries. Of course, the ImageNet moment itself, or the AlexNet moment that launched the deep learning revolution in the space of computer vision. So many people are comparing now this breakthrough of AlphaFold 2 to the ImageNet moment, but now...
So without getting too deep into the weeds here, AlphaFold 2 is an iteration on top of AlphaFold, which they have been trying to use to solve the protein folding problem. We'll watch a video on that in a second, but really we're just going to skip to the meat of it, which is like proteins and amino acids. So we already got kind of a talking on this about previous videos, but they all tie together.
Let's talk a bit about proteins and protein folding, why this whole space is really fascinating. First of all, there's amino acids, which are the basic building blocks of life in eukaryotes, which is what we're talking about here with humans. There's 21 of them. Proteins are chains of amino acids and are the workhorses of living organisms, of cells. And they do all kinds of stuff, from structural to functional. They serve as catalysts for chemical reactions. They move stuff around. They do all kinds of things. So they're both the building blocks of life and the doers and movers of life. Hopefully I'm not being too poetic. So protein folding is the fascinating process of going from the amino acid sequence to a 3D structure. There's a lot that could be said here. There's a lot of lectures on this topic, but let me quickly say some of the more fascinating and important things that I remember from a few biology classes I took in high school and college. Okay, so first is there's a fascinating property of uniqueness that a particular sequence usually maps one-to-one to a 3D structure. Not always, but usually. To me, from an outsider's perspective, that's just weird and fascinating.
I will say the same phenomenon exists in science. There's an enormous number of symmetries that we see in things, and it just seems to be without saying it's just magic, it kind of is just like a magic of the underlying nature of the world, that it has these natural symmetries.
The other thing to say is that the 3D structure determines the function of the protein. So one of the correlators of that is that the underlying cause of many diseases is the misfolding of proteins.
So this is the important part, right? Is we have DNA that is sending messenger RNA down into the cell. The ribosome is taking these amino acids and folding them into, it's not folding them into proteins. It's turning them into what they call polypeptides. And then the polypeptides are then sent out to be folded. So
now back to the weirdness of the uniqueness of the folding. There's a lot of ways for a protein to fold based on the sequence of amino acids. There's, I think, 10 to the power of 80 atoms in the universe. So 10 to the power of 143 is a lot. And you can look at Leventhal's paradox, which is one of the early formulations of just how hard this problem is and why it's really weird that a protein is able to do it so quickly. As a completely irrelevant side note, I wonder how many possible chess games there are. I think I remember it being 10 to the power of 100. Something like that. I think that would also necessitate removing certain kinds of infinite games. Anyway, off the top of my head, I would venture to say that the protein folding problem, just in the number of possible combinations, is much, much harder than the game of chess. But
so what's interesting about this is, hold on, I'm just taking some notes because I'm like thinking out loud here. The interesting thing about AlphaFold is that it has the potential to give us an insight into the building blocks of like our cellular structure that we have never had before, potentially come up with new ways of folding proteins. And if we can know that we have a particular protein that has a 3D structure that we need to do a particular activity, we can then code that back to mRNA. And we can put that in a vaccine or an injection. We can put that into our body, and then our body will then encode that mRNA into the little machines that we want and go after whatever the disease or, trigger the immune response or whatever it is that we're looking for to happen in this particular case. So in terms of that, we're not going to get too deep into AlphaFold itself, but one of the cool things about this is that the X-ray crystallography is incredibly expensive and takes a really long time, right? And it's quite the process. So if we actually can look up, let's go to YouTube and go X-ray crystallography. Let's watch a very quick video on it.
We're here today to talk about X-ray crystallography. I'm here with Mike Glazer, Emeritus Professor of Physics at the University of Oxford. He's going to be talking us through crystallography from crystal all the way through to model. So, Mike, if we can start with the crystal, you brought that one with you today, so it'd be great to hear about why this is special.
Okay, so this is a very large crystal, but you can see immediately it has nice flat faces, it has symmetry, and the reason for that is that we know that comes from the fact that the atoms and molecules in here are arranged in regular arrays throughout this crystal. So what we want to do in X-ray crystallography is to start with a crystal. We usually start with a much, much, much smaller crystal than this, and use a technique called X-ray crystallography to get to a picture of where the atoms are in the crystal.
So you would take a tiny sample, much, much smaller than this, and you would put it into a machine like this. So this is an X-ray crystallography camera, and you'd put this into the very center of this machine and have an X-ray come through here, down this tube, and hit the crystal. You'd put photographic film into this canister here, and you would then let the light reflect off the crystal all over the X-ray film, and you'd be able to move this in lots of different directions to collect lots of data about the crystal and how the light moves through it. And then from that data, you can do amazing wizardry mathematics, and then you end up being able to reverse engineer what the crystal will look like.
That's pretty well it. So what happens here is that we can, with a motor here, we can oscillate the crystal backwards and forwards around that axis. And then when we develop the film, we see spots on the film.
Okay.
Stay at home.
God, I've been muted this entire time. I'm so sorry, guys. Fuck the stream. This is what I got for not being prepared. The stream is just an absolute disaster. I might end up calling it early. We'll see how this goes. So, I ended up having another video queued up that we'll watch in a second. Before we get to that, the fascinating thing is that AlphaFold can give us the instructions for, the kinds of proteins that we need to build and how to fold them. We can then reverse engineer that technology and apply that using mRNA vaccines. Okay? So we talked a little bit about the cell, how the cell takes DNA from the nucleus, unzips it so that some enzymes can come in and expose that particular gene. A piece of mRNA is then created or transcripted off of the DNA, and the DNA closes back up, floats away. The mRNA is taken up into a ribosome, in which it is turned into a polypeptide, which is the first part of a protein, and then the polypeptide is folded, right? And this is, again, where the insight from AlphaFold is. So putting all of these technologies together, all of a sudden, we have a potential recipe for solving and curing a wide swath and variety of different diseases, which is why the research in these areas are incredibly exciting. So I'll just let Lex talk a little bit about why this is exciting, why is this breakthrough important, and then we'll dive into some more videos around mRNA vaccines.
Okay, why is this breakthrough important? Allow this computer scientist, AI person, to wax poetic about some biology for a bit. So because the protein structure gives us the protein function, figuring out the structure for maybe millions of proteins might allow us to learn unknown functions of genes encoded in DNA. Also, as I mentioned before, it might allow us to understand the cause of many diseases that are the result of misfolded proteins. Other applications will stem from the ability to quickly design new proteins that in some way alter the function of other proteins. So for treatments, for drugs, that means designing proteins that fix other misfolded proteins. Again, those are the causes of
The
long-term or the super long-term future impact of this breakthrough might be just the advancement of end-to-end learning of really complicated problems in the life sciences. So protein folding is looking at the folding of a single protein. So being able to predict multi-protein interaction or protein complex formation, which even in my limited knowledge of biology.
So when we were looking at this video before, right here, right? This is actin. It's a common protein inside of your body, okay? These all represent different molecules, atoms and their bonds that form these particular shapes and the properties of this molecule, right? It's incredibly complex. It is massively, these are incredibly, like when you look at it, it's like a mini universe that's happening at this level, right? So our ability to map out just the structure of these molecules is one thing. Being able to understand how they actually interact with each other inside of our bodies is a whole other, because there's some things that are too small for us to see with microscopes, so we just can't see how they actually work. It's not like we can stick a smaller and smaller microscope, because at a certain point light it just becomes ineffective at revealing itself. That's why we use electron microscopes at certain levels, because you can't really bounce light off of shit anymore. You have to actually bounce like tiny electrons, which are smaller than photons. So being able to point an AI at how these things interact with each other and give us insight would completely change the game in medicine and potentially give us the ability to come up with radical treatments to solve all kinds of diseases and really alleviate a lot of pain and suffering from the world. So it's an incredible mix of possible technologies that we could blend together. Let's look at mRNA vaccines really quickly. Hopefully it kind of brings everything that we were talking about.
Getting it ready to fight disease-causing organisms called pathogens. A vaccine is introduced to the body to mimic infection, triggering the body to produce antibodies against the pathogen.
Now, we went through the Kirksgezalt immunity video. We'll watch that at the end of this, kind of as a recap for everyone, for those of you who missed it last week, 'cause it's really important to this whole tying all this stuff together. Tying back immune responses, proteins, what proteins are in your body, how they're created.
But without causing the illness. Conventional vaccines usually contain a weakened or inactivated pathogen, or a piece of a protein produced by the pathogen.
Like polio. So the polio vaccine had inactivated polio in it. And there were kids who got fucking paralyzed because they got a bad batch of polio vaccine that hadn't been properly inactivated, right?
Called an antigen. RNA vaccines are a new generation of vaccines. Instead of the antigen itself, RNA vaccines contain a messenger RNA, mRNA, that encodes for the antigen. Once inside the body's cells, the mRNA is translated into protein, the antigen, by the same process the cells use to make their own proteins. The antigen is then displayed on the cell surface where it is recognized by the immune system. From here, the sequence of events is similar to that of a conventional vaccine. Some RNA vaccines also contain additional mRNA coding for an enzyme, which, after being translated in host cells, can generate multiple copies of the antigen encoding mRNA.
Nice.
This essentially amplifies the production of antigen from a small amount of vaccine, making the vaccine more effective. These are called self-amplifying RNA vaccines. RNA vaccines are easier and safer to produce than conventional vaccines. This is because mRNA molecules can be synthesized in a cell-free system using a DNA template with a sequence of the pathogen. While conventional vaccines usually require a more complicated and risk-prone process of growing large amounts of infection,
They actually have to culture the virus. They have to grow the virus or grow whatever it is that's going to get them that antigen. They have to culture it in a large quantity, and then they have to process it in a way in which they can get it into the body. So
infectious pathogens in chicken eggs or other mammalian cells. Without the risks of being contaminated by infectious elements or allergens from egg cultures, RNA vaccines are also safer for patients. Because protein synthesis occurs in the cytoplasm, RNA molecules do not need to enter the nucleus, so the possibility of them integrating into the host cell genome is low. RNA strands are usually degraded by cellular enzymes once the protein is made. The relative simplicity of the production process makes it easier to standardize and scale, enabling rapid responses to emerging pandemics.
yeah, let's be real though. The drug companies need to let up on the patents though. If companies like Pfizer and Moderna are not going to make the technology readily available to other countries, then talking points like this are absolute bullshit. Sorry. Real talk alert.
Other advantages include lower production costs and the ease of tweaking RNA sequences to adapt to rapidly mutating pathogens. On the minus side, it can be challenging to deliver mRNA effectively to the cells, since RNA sequences and secondary structures may be recognized and destroyed by the innate immune system as soon as they are administered intravenously. These limitations can be overcome by optimizing codons, using modified nucleosides to avoid recognition, and packaging RNA into protective nanoparticles.
This is the cool part. This is the part that they were like, holy shit, we figured something out here. So we, for a long time, have thought about the idea of using messenger RNA to deliver proteins into the body. That's not necessarily a new idea. The breakthrough was being able to figure out some of the optimization and being able to package it, what they call a lipid or a fat molecule. So they're able to synthesize this tiny little lipid nanoparticle that encompasses or envelops the messenger RNA. So they can deliver it into the cell. It can get in the cell just far enough so that it can replicate, create the protein that it needs to, and then degrade appropriately.
Particles. Another disadvantage is that most RNA vaccines require uninterrupted refrigeration for transportation and storage, which can be a hurdle for vaccine distribution. Research is ongoing to engineer thermostable vaccines.
So I would like to go back to Friedman. And he was talking to this guy. We're going to pick up kind of where we left off last time in their conversation around
I'm thinking, like, there's got to be long.
how does the mRNA vaccine work. So we're going to recap on that. And it'll get into sort of like, you know, are they safe? So let's start here.
But, you know, I still think it's very surprising that something like that works, right?
Yeah, I am surprised.
So you're just launching RNA in a protective membrane.
Sup,
Yeah.
extra more and Affinitos. Thanks
Now,
for coming to say hey if you guys are
One thing
there.
is surprising that the RNA sort of lasts long enough, right, for
That's
in its structure. But then the other thing is, why does it work that's a good training ground for the immune system? Is that obvious?
Well,
that
I
should
don't
work?
think it's obvious to most people, and it's worth going into because it's really interesting. I mean, first of all, they wrap the RNA in fats, in lipid membranes, right? And the particular formulation, they test for years to make sure it's stable, you know, it lasts a long time after it's injected. And the two companies that make the current COVID vaccines, right, Moderna and Pfizer, they have different lipid formulations to get to the same. So that's a real part of it. And it's not simple. There are quite a few different lipids that they put into this coating. And they test to see how long they protect the RNA after it's injected, say, into a mouse. How long does it last? And the way it works is these lipid nanoparticles, they get injected into your muscle, they bump into cells, and they get taken up. So lipid fat is sticky. It's greasy, we like to say. And so your cells are covered with a greasy membrane also. So when these lipid nanoparticles bump into them, they stick and they eventually get taken up. And they figured this out right at the beginning. If we put RNA in a lipid nanoparticle, will it get taken up into a cell? And the answer was yes. It was just, let's try it, and it worked.
So it's basically experiment. It's not like
pretty
some
cool
deep understanding of biology. It's experimentally speaking, it seems to work.
Yeah. Well, they had some idea that lipids would target this to a cell membrane. And remember, there's no receptor involved. Like the virus has a specific protein that attaches to a receptor.
Right.
It's not efficient enough to just bump around and get into a cell. That's what these things are doing, and they probably optimize the lipids to get more efficient uptake. But it's not as efficient as a virus would be to get into a cell.
So you have no specific. I mean, which is why it's surprising that you can crack into the safe with a hammer or with some fat. I mean, that's kind of surprising. It's kind of amazing that it works. But so maybe let's try to talk about this. So one of the hesitancies around vaccines, or basically around any new technology, is the fact that mRNA is a new idea, and it's an idea that was shrouded in some skepticism, as you said, by the scientific community, because it's like it's a cool new technology, surprising that it works. What's your intuition? I think one nice way to approach this is try to play devil's advocate and say both sides. One side is why your intuition says that it's safe for humans, and what arguments can you see, if you could steelman an argument why it's unsafe for humans, or not unsafe for humans, but the hesitancy to take an mRNA vaccine is justified.
So many people are afraid because it's new technology and they feel it hasn't been tested. I mean, in theory, what could go wrong? This is the nice thing about mRNA is that it doesn't last forever, as opposed to DNA, which doesn't last forever but can last a lot longer, and it could even go into your DNA, right? So mRNA has this shorter lifetime, maybe days after it's injected into your arm, then it's gone. So that's a good thing because it's not going to be around forever. So that would say, okay, so it's sticking around for your lifetime is not happening. But what else could happen? Well, let's see, the protein that's made, could that be an issue? And again, proteins don't last forever. They have a finite longevity in the body. And this one also lasts perhaps at the best a few weeks.
Now, this is a protein that's made after the RNA gets into the cell.
Yeah, so the lipid nanoparticles taken up into a cell, and the mRNA is translated, and you get protein made.
And there's also a question, and I'm sorry to interrupt,
Yeah,
like where in the body, so because it's not well targeted, or I don't know if it's supposed to be targeted, but it can go throughout the body. That's one of the
Right,
concerns.
so it's injected deep into your deltoid muscle, right, your shoulder. And the idea is not to put it in a blood vessel, otherwise it would then for sure circulate everywhere. So they go deep in a blood vessel, and it's locally injected. And they did, before this even went into people, they did experiments in mice where they gave them 1,000 times higher concentrations than they would ever give to people. And then when you do that, it can go everywhere, basically. You can find these nanoparticles in every tissue of the mouse. but that's at 1,000-fold higher concentration, right? So I think at the levels that we're using in people, most of it's staying in the muscle, but sure, small amounts go elsewhere.
Could there be a lot of harm caused if it goes elsewhere? Like, let's say ridiculously high quantities. I'm trying to understand what is the damage that could be done from an RNA just floating about.
So this is an important conversation to have, right? Is we should actually talk about, like, what are the possibilities of something in this case going wrong? You know, like, would it require like an insane dosage that is nothing close to what you're getting in real life? Like, there's a whole. It's better to understand the boundaries of what can go wrong and have open conversations about that than not have those conversations, in my opinion. So I'm glad they're talking about this.
So the RNA itself is not going to be a problem. It's the protein that is encoded in it, right? This is a viral RNA which has no sequence in us, so there's nothing that it could do. It's the protein that I would say you could ask, what is that going to do? And the one property we know about the spike is that it can cause fusion of cells, right? That's how the virus gets in the beginning.
Right.
The spike attaches to the cell by this ACE2 receptor, and it causes the virus and the cell to fuse. And that's how the RNA gets out of the particle.
But so, wait, I'm a bit confused. So with this mRNA vaccine, with the lipids and the RNA, there's no spike, right?
The mRNA codes for the spike.
Oh, the mRNA codes,
Yes.
so it creates the spike.
Creates a spike.
And so that spike could cause fusion of cells.
Yes, except they modified the spike so it wouldn't.
Got it.
They made 2 amino acid changes in the spike so it would not fuse.
So they understand enough which amino acids are responsible for the fusion.
That's right.
Was the idea, yeah. So they try and engineer in a
So now it's not going
they try and engineer in a way that doesn't actually cause like inflammation, all these other issues, right?
to cause fusion, so that's not an issue. It's called the pre-fusion stabilized spike.
Cool.
So the spike, when it binds ACE2, that top falls off, and the spike, and the part of the spike that causes fusion is now exposed, and that doesn't happen in this mRNA vaccine. So those are the things that could have happened, but I think they're ruled out by what we've just said. But there's no better test than putting it into people, right?
Right.
And doing phase 1, phase 2, and phase 3 and increasing numbers of people and asking, what do we see? Do we have any concerns? And so now it's been in many millions of people, and we don't see most of the effects you see in a vaccine, you see in the first couple of months. Things like the myocarditis with some of the vaccines, the clotting issues with the AstraZeneca vaccine, Guillain-Barré, you see those relatively quickly. And we've seen small numbers of those occur, but other things we haven't seen, and you know, you never say never, right?
Right. So I mean, this is fascinating, right? It's like I drink, I put Splenda in my coffee, and has supposedly no calories, but it tastes really good. And despite what like rumors and blogs and so on, I have not seen good medical evidence that it's harmful to you. But it's like, it tastes too good. So I'm thinking like there's got to be long-term consequences, but it's very difficult to understand what the long-term consequences are. And there's this kind of like distant fear or anxiety about it. Like, this thing tastes too good. It's too good to be true. There's no free lunch in this world. This is the kind of feeling that people have about the long-term effects of a vaccine. That you mentioned that there's some intuition about near-term effects that you want to remove, like the diffusion of cells and all those.
Okay, so here I think it's a really, really important point to tie together, and I think maybe where people like Lex, I'm not accusing him of this, but are kind of like losing the plot, right, is how the immune system works. So the entire idea behind providing someone with a vaccination is to provide a long-term immune response, like or a long-term T cell that is going to encode